Large marine mammals are some of the most polluted animals on the planet. This is a result of a process known as biomagnification, where chemical pollutants get ingested by small marine organisms and build up in the fatty tissues of larger animals as they move up the food chain. Animals at the top of the chain, such as killer whales, can end up carrying over 100 times the harmful level of pollutants, enough to classify them as toxic waste. Chemical pollutants can have deadly effects on the health and survival of these animals, making them a major threat to these species.
Well, we know that these chemicals can impair the animal's ability to reproduce and to handle infections and disease. What we know much less about are the specific internal mechanisms that cause these things to happen.
Studying the effects of pollution on marine mammals is a challenge. These animals are widely distributed across the world's oceans, so finding them can be difficult. In addition, scientists need samples from organs like the liver, kidneys, and gonads. These are places where pollutants tend to build up, but getting organ samples from wild animals is nearly impossible. How do you study the effects of pollution on large marine mammals in a way that's both humane and effective?
The Marma-detox project is an international research project that aims to address all of these challenges using cell cultures. Our goal is to understand the effects of ocean pollutants on large marine mammals in a way that is efficient for the researcher and ethical for the animal. To do this, we partner with another research project called SLICE, who takes small skin and blubber biopsies from whales in the wild. We then use skin pieces from those biopsies to create cell cultures. We can use these cells to study effects of pollution in marine mammals in ways we never could before.
Marma-detox and SLICE are sister projects in that we are both trying to understand the specific effect of chemical pollution on large marine mammals. In the SLICE project, we want to focus on the tissue level by keeping alive blubber slices to further expose them to pollutants and stress hormones. To obtain our sample, we use DART to take small biopsy from wild whales, just below their dorsal fin. So far, we have been able to collect biopsy from fin whale, humpback whale, and killer whale. After we collect the biopsy, we store them in a sterile container and ship them to the lab for experimentation.
Our combined labs are currently conducting 3 different types of experiments. So the first is the isolation and establishment of the whale cell cultures, which is done by the Marma-detox. So we grow the cell cultures and then expose them to different levels of mixtures of pollutants to study how these mixtures affect the cell's DNA and cellular processes.
So here we are growing killer whale cells, fibroblasts, from skin biopsies, and they're looking good.
SLICE conducts experiments to test how pollutants affect large marine mammals at the tissue level. So they do this by looking at how pollutants affect groups of different cells, rather than just one cell type, like in the Marma-detox.
We all know that pollution has very bad impacts on the environment and mammals in general, but actually we don't really know the specific effects of pollution. So we would like to know what's going on and in which way we will be affected by pollution. So our project will focus on doing transcriptomic analysis and to try to better understand which genes will be increased in terms of transcription or decrease due to the exposure of pollutants. Thanks to this insight, we might be able to take actions to try to better protect those threatened mammal species and hope for a better future.
So once the biopsies arrive at the lab, they immediately cut these into very thin slices of blubber. So in the SLICE project, the slices are exposed to the mixture of pollutants and cortisol, which is a type of stress hormone. And they do this to study how the slices respond to a combination of both pollution and stress. So Marma-detox and SLICE are together optimizing the usage of each biopsy, where SLICE uses the blubber for slicing, and Marma-detox uses the skin part for establishing cell cultures. So the nice thing about our research method is that we can obtain our samples without bothering the whales, well, too much. While the downside of this method is that the range of cell types we can obtain is limited to the cells found in skin and blubber tissue. So these cells don't express all the response pathways we want to study. Ideally, we want to study how pollutants are affecting organs like liver, brain, and fat tissue, for example. But these are tissues we can only obtain from dead whales. So instead we are trying to reprogram the cells we have into other cell types, such as liver cells and fat cells. We might even be able to reprogram them into brain cells with some help of collaborators in Italy. I mean, if we're successful with this process, the research possibilities will be endless.
The biggest advantage about working with cell cultures is that these cells can be maintained and multiplied to large numbers in the right conditions. The methods we try to develop will make our research much more convenient and affordable than if we were to study these animals in the wild. This kind of research is difficult to perform, and you need the combined resources of several partners. So we are very excited to be paving the way for toxicology research on marine mammals.
Marma-detox and SLICE are projects that could potentially play a key role in helping to create cleaner oceans. The findings from their research will help us understand and beat marine pollution, a major challenge that the UN is looking to address by 2030 through their Decade of Ocean Science for Sustainable Development. By providing important knowledge about how pollution affects marine mammals, Marma-detox and SLICE can help create solutions and policies that would reduce or ban chemical pollutants from entering the ocean. Through their innovative research, the Marma-detox and SLICE projects hope to build a more sustainable future for our planet by supporting healthier marine mammal populations and a cleaner ocean for us all.